Okay, I'm going to walk you through how to interpret the results of your electrophoresis gel. So just to recap, we collected DNA from our cheek cells. We mixed that DNA with a PCR master mix, and that contained primers and polymerase, Taq polymerase, and nucleotides. We mixed that together. We put it in the PCR machine, which heated and cooled the mixture, in our case 40 times. And when we take the little PCR tube out of the PCR machine, it doesn't look like it has anything in it because the DNA is invisible. We have to load the sample, the whole volume of the sample, onto a gel. So we put it in the well. We mix it with a little loading dye and put it in the well. We also have to put in the well something called a size standard or a marker or a ladder. All of those terms are sometimes used. In this case, the ladder, the marker is in lane 1, and this gel's already been run and stained. So now we can see the DNA on the gel. And they've gone ahead and marked the sizes of these bands in the size standard lane, and you can use them to confirm that the size of bands you got on your student samples matches what you expected. So this is the results. This is it. What you're going to do, though, is to look at the results and make reasonable conclusions. So results are just, there are bands in the lanes. Some lanes have 2 bands, some lanes have 1 band. You know, something that anybody could see even if they didn't have any training in genetics. But the conclusion, what this probably means, takes some knowledge of PCR, it takes knowledge of electrophoresis to understand what this probably means. And so you're going to be asked to make conclusions. So let's say that lane 2 and 3 and 4, let's say those were controls. So we had some DNA that we knew is homozygous with the ALU insertion. And so what in this, let's see if I can get a pen going here. In this system that we're using, they're calling something that's homozygous for the insertion, they're calling plus. So plus over plus would be homozygous for having the insertion. So this DNA came from somebody who is known to have a homozygous ALU insertion on both copies of 16. 3 would be the control that is, minus over minus, and they only produce the smaller fragment, which has no ALU on either copy of 16. And number 4 looks like it's the positive over minus control, and what that means is that one copy of 16 has the ALU insertion and gives the longer fragment. One copy of 16 gives the shorter fragment. So both copies are in the test tube, so both copies will be amplified by PCR. And so you can see here on the size standard lane, this is 1,000 base pairs. So if we're estimating that our longer PCR product is about 941, you can see that it's just a hair below 1,000, so that's reasonable. And then the other one is supposed to be 641, and you can see that this one is somewhere in between the 5 and the 7 on the marker, so that's a reasonable size. Those look to be the proper PCR products, nothing unexpected. You do see some things here. Sometimes these are called non-specific or heteroduplex. There are some other bands that we're not going to worry about since we're just beginning our interpretations of this. If you were really needing this information, some of this you would learn how to interpret, but let's go with the really dark bands, okay? So on this, so these are your controls. So then these are your students. This is Anna and Barbara and Chandra and Derek. I don't know. Student A, student B, student C, and student D. And so you're going to make some conclusions. Conclusions are going to be genotypes because you can't see the genotype. You can just see a band of a certain size, but it's going to strongly indicate a certain genotype. So student A, the conclusion is student A appears to have a band, a single band that corresponds with having a homozygous negative, well, homozygous no ALU insertion genotype. That would be a conclusion. That's the most likely conclusion. Student B seems to have a genotype that corresponds to homozygous insertion. So both copies of 16 have the insertion. Student D also appears to be homozygous negative. Student C, this faint band here is probably real, and I only know that because I've done a lot of these, but student C, I guess if you put he could be hetero or he could be minus over minus, I would accept either one of those. I think he's hetero, but I'm going to grade this based on, you know, what a student who's just starting out would reasonably think. So that's how you're going to interpret these. You really have to be given the fragment sizes of your size standard lane. And remember that the smallest fragments run the fastest and the largest fragments run the slowest. So that's how you would interpret the genotypes of these 4 students.